Almost 1 in 5 people will suffer from some form of mental health problem in their lifetime, making it the leading cause of disability worldwide. The main problem is that no new drugs have been discovered in decades, and many patients do not respond to current clinical treatments. What's more, doctors can't access live brain tissue as easily as other parts of the body. Researchers led by a team at the University of Cambridge have developed a new method to discover drugs for psychiatric disorders. They use blood cells donated by patients with schizophrenia and healthy individuals as a model for discovering new drugs.
Increasingly, there's evidence that psychiatric disorders affect not only the brain but also the rest of the body, particularly the immune system. Inflammation processes and metabolic changes have been associated. So therefore, the scope of our group was to investigate whether we can identify abnormalities in peripheral blood cells for psychiatric disorders.
The team uses a combination of laser-based flow cytometry for in-depth analysis of single cells, a unique toolbox of fluorescent dyes to label structures inside the cell, and robotic sample preparation to incubate the blood cells with thousands of chemical compounds. This allows the team to identify new drug targets and potential drug candidates for the treatment of schizophrenia.
So, what's new about this study is that instead of just measuring genes and proteins, we explore live cellular function as a model of disease. And to do this, we use a technology called high-content screening. And this enables us to measure thousands of behaviors within the cell at the same time. And we use cells directly from patients because we believe this gives us a better idea of the disease in its native state.
Specifically, the team have focused on finding new psychiatric uses for existing drugs, which are routinely prescribed for other conditions such as high blood pressure.
This drug repurposing strategy can reduce the time and cost it takes to bring a new drug to the clinic. It represents an efficient alternative to deliver novel treatments to patients in less time.
The team have also used the approach to test existing psychiatric treatments on patient blood cells and predict how effective those treatments will be for each individual.
The problem with schizophrenia is that many patients don't respond to first-line psychiatric treatment, so it's a trial and error. One has to try out several medications before they may or may not respond. So it would be very important to identify patients who will respond to a certain drug ahead of the time so that they don't have to go through this trial and error period, which may last months and sometimes years.
As a final step, the team confirmed that the activity of new drugs was shared between blood cells and brain cells by testing those drug compounds on human nerve cells. Although the research is in initial stages, the team is hopeful that this approach may eventually lead to new drugs for psychiatric disorders and better treatment choices for each individual patient.
This study has the potential to substantially accelerate drug discovery and personalised medicine for neuropsychiatric disorders, but also other human diseases.